So our next speaker is Joakim Dillner, who will address a very important question of how this vaccine actually works.
Thank you very much, John. I'm Joakim Dillner, and I was asked to speak on the question of how do the vaccines, how do they work? And I assume that when the organizers ask me, an antibody buff like me to talk about that, they assume that the talk would be mostly about antibodies, and this is indeed the case. My conflict of interest statement is that I've acted as a consultant and received research grants from Merck and SBMSD. The antibodies to the L1 VLPs, they are known to neutralize viral infectivity and known to efficiently protect against the type-specific infection in animal model system. Already since many years has this been known, and it has been formally proven that the vaccine-mediated protection in animals is humoral by the serum transfer experiments. You can take the serum from animal and transfer the immunity to the next animal. There is also a correlation between the protection and if you have neutralization activity in the serum. This has been shown in several animal model systems, first in the cottontail rabbits and then also in dogs and rhesus monkeys. This is just an example of some of the original studies on this, where you could see that when you immunized the rabbits, they got very high antibody titers that were neutralizing and that protected from challenge, and then this transferred to the non-immunized rabbits that transferred the immunity to them. For humans, it has actually for quite a long time been known that the antibodies that are induced by infection are indeed also neutralizing. It has been known for 20 years. And it's also well known that there is no correlation between the antibodies and clearance or progression, and there appears to be no therapeutic effect of the antibodies. And it's also known that the IgG antibody response to the VLPs is highly stable over time in the infected person at least for 15 years, has been demonstrated in follow-up studies, which should be good news for the longevity of the protection as well. Now, the neutralizing antibodies that are induced by vaccination with VLPs, they are known to block the infectivity of the virus, and they have these type-specific neutralizing antibodies that bind to the VLPs. There is cross-neutralizing activity of antibodies, but it's typically 10 to 100-fold less in its neutralizing activity compared to the type-specific neutralization. And the reason for the high type specificity is that the antibodies primarily react with surface-exposed hypervariable loops on the virion surface. And actually most of the particle surface of a papillomavirus is covered with such hypervariable loops that are different between the different papillomavirus types. This is a schematic view of how the papillomavirus particle is built up. You start with the L1 molecule that spontaneously self-assembles in 5 copies to the capsomere, and then into hexavalent capsomeres, and then they build up the entire virus particle like that. The L2 protein, the little green dot, is situated in, so to say, the middle of the capsomeres here. I will come back to the L2 protein in a few seconds. This is a side view of a capsomer with the hypervariable loops shown in colors here. And the top of this figure is the outside, directed outwards from the particle. And you can see that actually the hypervariable loops, they cover almost the entire top surface of the virus. And on the inside of the particle, you have these residues that are highly conserved between the different papillomaviruses, but they are on the inside and are not reacting with the antibodies and are not giving a functional reactivity. But they are more to say that the epitopes on the inside of a particle are more like a a problem when designing assays on how to measure the type-specific and neutralizing antibodies, because particularly if you vaccinate with disrupted or not really intact VLPs, you may have antibodies also against internal epitopes of the virus. Now, why do we need assays to measure HPV antibodies? Well, it's clear that we will need to have continuing clinical trials globally for a number of reasons. They will be essential for promoting the global implementation of HPV vaccines. We will have both so-called implementation trials in phase 4 and 5. It will be anything from quality control of new batches, trying new target groups like the HIV infected or the infants or small children. And there could be logistic improvements such as getting rid of a cold chain or other ways of administrating the vaccines. For all of those, we will need some immunogenicity evaluation of such trials, because it's not realistic to study these minor changes with clinical disease as an endpoint, and it's not really realistic even with an infection endpoint either. So one would need to have an immunogenicity endpoint for those. And of course, second generations vaccines. We are all hoping that we will have less expensive and more broadly cross-reactive second generation vaccines upcoming. And for the whole area of trying to optimize those in the basic research, you would need reliable immunogenicity measurements. Immunogenicity is already becoming an increasingly important matter of discussion when it comes to thinking about trial endpoints. As Susanne just mentioned, there has been discussion now for more than 10 years on whether one needs clinical endpoints in trials or whether looking at a persistent infection as an endpoint would be enough, which there are very good arguments for. However, with the present situation with very effective vaccines that are highly protected against the infection, it starts to be an ethical problem even to use infection as endpoint in the trials, and that one may need to resort to evaluation of using immunogenicity. Of course, everyone knows that when we define the susceptible populations in trials, they should be DNA negative and HPV seronegative to ensure that we are naive to start with, which is a basic thing to at all look at trials. And then, of course, if we don't have standardized antibody assays, the non-standardized definition of both trial endpoints as well of the susceptible populations that go into the trials, different rates of disease and different efficacy, and no comparisons possible for any sort of improvement or between different vaccines. Just a few words on the different types of assays. This is the most widely used test. That's the virus-like particle-based ELISA. It's simple, rapid, it's reasonably reliable, and it's widely used. It measures binding antibodies, but the binding antibodies are, although not functional in a biologic sense, these are strongly correlated to neutralizing activity. And they are dependent, if you are going to do reliable virus-like particle ELISAs, you need high-quality VLPs that expose only the type-specific surface epitopes in your ELISAs, as low-quality VLPs may expose the internal non-neutralizing and broadly cross-reactive epitopes. As part of the WHO HPV LabNet, we have produced an HPV laboratory manual with a detailed standard protocol for such ELISAs and developed key international reference reagents, including an international standard that enables calculating international units of levels of HPV antibodies. And they can be ordered from the standard catalog for anyone. The important development here was the pseudovirus neutralization assay as invented by Patricia Day and John Schiller and colleagues already several years ago, where the papillomavirus particle is packed with a reporter gene where you can measure whether infection has occurred using an assay for for a protein generated by the reporter gene. In this system, it's the secreted alkaline phosphatase. And in this system, where the antibodies do bind the particle and prevent infection, it's already that you have come some way further from binding to actually measuring a biological activity. This was the first trial that we did in the global HPV LabNet, where we are trying to have a global quality assurance and comparability of HPV antibody measurements. This LabNet was an WHO initiative. It was funded between 2006 and 2011 by the Bill and Melinda Gates Foundation to promote development and use of HPV vaccines by international standards for HPV genotyping and HPV antibody assays. And the first international collaborative proficiency study of human papillomavirus serology resulted in that we had 10 labs participating all over the globe, out of which 6 were proficient in the VLP ELISA. We had a very good correlation of the neutralizing activity to the HPV DNA status of the serum donors, as well as a correlation to the binding antibody levels that was, in principle, one can say that if there were binding antibodies at a level above 10 international units, all serum samples were neutralizing, and if it was less than 6 international units, none were neutralizing, and in between there it was not so good correlation. But in principle it's a strong correlation. Now, this is the current state of the art, but one should be aware of that this does have limitations, because neither the ELISAs nor the first generation neutralizing assays have managed to define a minimum antibody level required for protection. And there has been a very intriguing finding, particularly from the Merck trials on HPV-18, where the protection has been found to persist also when no detectable antibodies are present, implying that the assays are just not sensitive enough to go all the way to the minimum level required for protection. Another problem is, of course, that we have so few vaccine breakthrough cases that our studies are not well powered to look at the correlates of protection. Another limitation is the data from the L2 vaccine candidates that do show broad protection in animal model systems, but only low neutralizing antibodies in the first generation neutralization assays. And there is also a problem with this, and that is that L2 is not exposed on the particle, implying that there may be several steps in the infection and protective mechanism that the L2 antibodies against L2 are interfering with. And there we have had some developments in recent years, and it's an improved understanding of how infection and protection works. I think this is one of a very good example of how basic research can result in improved understanding of how infection really works, and advance the clinics into new assays and for the future. In principle, what's happening is that the particle binds to the basement membrane. There's a protein cleavage inducing a conformation and change of a particle, and the L2 is exposed and the receptor binding structure is exposed. Only after this is the attachment to the keratinocyte and infection occurs. This is very interesting because it explains the known requirement that the papillomavirus must reach the basement membrane in order to have an infection. So in this schematic figure, there are 3 mechanisms of protection described. If you have a high level of anti-L1 antibodies, the particle is so covered with them that it doesn't even reach the basement membrane. If you have a low level of anti-L1 antibodies, it does reach the basement membrane, but the low level of L1 antibodies in panel C here are sufficient to prevent it from binding to the keratinocyte receptor. And in panel D, we see the example of the L2 antibodies that will not bind with the intact particle, but they will be bound to the particle after it has bound to the basement membrane and has been modified and exposed to L2 such that it can't bind to the receptor. So when there has been recent reports of a second generation HPV neutralization assay where you sort of try to mimic these in vitro, where you generate an extracellular matrix in wells like this, add the particles to the extracellular matrix mimicking the basement membrane, and then you cleave them and adding the furin proteases, add the antibodies, and only that, after that, do you add cells that the virus can infect. And it's very interesting here that this has resulted in that there is a large improvement in sensitivity of detection of neutralizing antibodies in these type of second generation neutralizing assays. And it's sort of promising that we might be getting on the path towards more sensitive assays with biological relevance that could be used to define the correlates of protection. To summarize what I've just said, the preventive vaccines, they work via induction of neutralizing antibodies. If there is high amount of antibodies, they prevent binding of a particle to the basement membrane. Low amounts of antibodies prevent binding of a particle to the keratinocyte. The antibody measurements can be reported in international units, as this has been standardized and really should be part of every study of antibodies nowadays. And we have an example how improved understanding of a mechanism of protection has allowed design of improved neutralizing antibody tests, although I should say that no assay has been shown to correlate to the protection for humans so far. Thank you very much.